H2 and NADH. And then it consumes oxygen, it makes water. The enzyme that it uses is ATPase. So in that case, I have mentioned ATPase in the LT assignments. If you haven't seen those, they are. I mentioned it last week, and I just mentioned it today. I don't know. Do you think ATPase is a good chance of being on the exam? And more immediately on the quiz tomorrow? I'm not sure exactly what each of you are going to get, but it's definitely in the list of questions. Okay? So once again, cross-reference what we did in lab, when I wrote the notes out, what we did in lecture, what I'm about to do in lecture, what we do in lab on the LT assignments. And if you see that 3 times, study that stuff. If you see it once and I'm like, it's not really that important, but I'll talk a little bit about it, you might say, I'm not even gonna forget. I'm gonna forget about it because it's gonna be 1 or 2 questions. Okay? So trying to figure out and anticipate what's important to the lecturer is really, really a critical strategy or skill. I remember a long time ago, I was in class with a friend of mine, and he and I were studying genetics. And I go, Dan, here's how we do it. He goes, what are you, dumb? That's not how you do it. You do it this way. And I said, no, you're wrong. And then I looked and I was like, he's right. So anyway, so now he and I are studying, and he shows me how to do this exam, this question. We get to the exam. He anticipated it perfectly what the question was going to be. Almost that exact question was on the exam. And I was like, it was a little bit changed. Things were changed a little bit. And I was like, hey, in the middle of lecture exam, I was like, damn, number 2. And he's like, deer in the headlight. Because he saw, he knew the stuff, but he saw the differences in the questions. And I saw the similarities in the questions. Because they were different for him, he got it wrong. Because I saw the similarities, I got it right, or I almost got it right. So that's another thing, is that once you start anticipating what you need to know, if I ask it slightly differently, look for the similarities. If it's talking about glycolysis, I might talk about glycolysis in the context of something and then ask about what type of chemical reaction. Right? I might talk about, which would be chapter 2. I might talk about glycolysis and talk about the next steps, which would be the cell cycle, right? Or the cell. So, anyway, are there any questions about that? So anticipating what's going to be on the exams and our quizzes, which leads me to this thing right here. So here's our schedule, broadly stroked, right? So today's, the only thing that I'm going to do today is talk about chapter 3, and we'll finish it today. If something catastrophic happens and we don't get through, then whatever we don't get through will not be on this quiz. Tomorrow we'll have a quiz, quiz 4. It'll be 30 questions just on chapter 4, no, chapter 3. And we'll do that here. After that, we'll start on talking about tissues. Tissues is chapter 4. And we'll finish that on Monday and Tuesday. It's sort of a long lecture. You guys also have to balance multiple things. You have to walk and chew gum at the same time. So don't just study the lecture notes. Remember that we have a mini practical on Thursday and Friday. Thursday morning is a large lab. We have to start at 8 o'clock. Okay? Thursday afternoon starts at noon. Friday morning starts at 8:30 because we have a smaller lab. Okay? And once again, if you're there early, then you can put your name on the list, wherever you want. But then you have to remain in class until the until the mini practical starts, or until the class starts. And once again, you guys remember V from last year? She and a couple other friends did this. They had a lab in the afternoon, and they came in early, put their names up on the list, and then they went out for lunch. And when I say lunch, they didn't go to the cafeteria, right? They went to the Providence Place Mall, right? And technically, I didn't specifically say don't do that, and so they did it, and I started laughing at them, and I made fun of them, and then I said, but don't do that again. So I didn't say it. Oh, what are you gonna do? So that's V's rule now. so that's the new thing, right? So you gotta be in class in order to keep your spot. Now, so that's the axial skeleton. Remember all the bones? And if you have questions about where to find that information, it's both in the lecture brightspace and the lab brightspace. And then we're rapidly going to next Wednesday, which is the exam, the first exam. What's the first exam look like? It looks just like the quizzes, except for the syllabus quiz. It looks just like quizzes 2, 3, and 4. There's gonna be 4 chapters. Generally, the first 3 exams will each have 4 chapters. And this is the distribution of questions. Multiple choice questions here, 25 in the first exam, first chapter, and 30 in each of the next 3. During that day, when we have exam 1, we don't do anything else. Generally ask me to do extra work with you guys after the exams, but I said no, because she's crazy like that. She's just joking. The only thing that you would do on exam day is come in, get online, and then take the exam. I'll open it up for each of you individually, okay? The exam will have, this exam only, you'll have that copy of the periodic table, and then I'll also bring extra papers if you wanted to, blank piece of paper if you want to jot things down. If you want to write things and test your memory and stuff like that. Like, I think it goes this, I'm not sure. Okay? So that when I do the quiz tomorrow, when I do the exam, I'm going to be very, very focused on exam integrity. Hold your hand up, your left hand. Say this. I'm going to be very focused on exam integrity. I'm not accusing her of anything like that she's cheating, but I'm going to make sure that you don't have anything in your ears. I'm going to make sure your phones are not on the desk, and I'm going to make sure you don't have any watches on. Even if it's an old-fashioned watch, I'm going to make you take it off. Okay? So I'm going to have you go like this, and if you want, you can go like that, you can go like that, right? Let's see. That's just for you. All right? So I'm going to make you go like this. I'm going to make you go like that. And if some of you have hair, I'm jealous. I might make you go like this and put your ear, show me your ears. And then I'll also say, put your phones away. Now, this, I'm sorry for picking on you today, right? So her phone's right here.
Mine.
Oh, it's yours. I'm sorry for picking on you then. That is not acceptable during an exam. Okay? It has to be in a bag, away from the desk, zipped up. By the way, mine's off. What should you do with your phone? Turn off notifications during an exam. Someone had something on, it was ringing last time, and it's annoying. Okay. Just make it a rule. You walk in this building and you turn off your notifications. So any questions about the schedule, about the structure of the exam? The reason why I give you so many quizzes, these 3 quizzes in a row, is that then you're going to get used to some of the questions that are going to, you're going to have a whole bunch of the questions, almost 3/4 of the questions that you've experienced. And so some of those questions, that same library is going to be used for the exam. So you're going to have some of the same questions repeated. Okay? All right, so any questions for me? All right, so let me see if this starts now. So while I'm waiting for things to start, I gotta text my wife.
Oh.
just did. And I asked about the dog. I always do that. All right, so let's talk. Let's see. I sort of wish that the professor from Rick just took this class over, because I don't really like this way this podium is set up. I was at URI over the weekend visiting my son, and one of his friends, as we were saying goodbye, one of his friends plopped down on the sofa. He's in a suite. Plopped down on the sofa, and I looked over his shoulder because his laptop was up, and this exact screen was up. And I said, "Oh, you taking A&P?" And he goes, "Yeah. How do you know?" And I said, "Well, I know this logo right here." He's an A&P too. He's a sophomore, and he was studying the eye. He picked up exactly where we left off, where we'll leave off in this class. So you guys will be able to go to URI I'm pretty sure to Rick. That's a little iffy, and then definitely to CCRI. The curricular aligns perfectly. Okay. Rick's a little off. They've been off for a while. They're slowly getting back to catching up to URI and CCRI. And it's not that Rick's bad or anything like that. It's basically, it's a couple of old old people that don't feel like changing.
Yeah, why is the graduation rate like 42%?
At Rick?
Yeah.
I don't know. That's beyond what I'm talking about. I'm talking about just people in the biology department that don't feel like changing. So I don't know.
Well, that correlates to it.
Well, right, it may very well be that too. All right, so there's learning objectives, and if you look at this list of things, I'm never gonna talk about it, but if you look at all the stuff that I possibly could be talking about, I could be talking about a whole bunch of stuff, right? So what are we gonna be talking about? Here's the next thing. This chapter 3 here, we're gonna be talking about the basics of the cell. Right, we're going to be talking about what's inside the cell and what's outside the cell. We're going to be talking about the plasma membrane and the cytosol. We're going to be talking about the nucleus and other organelles. There's a distinction between membrane-bound organelles and organelles that you can't draw or that are difficult to see when the cell is not undergoing normal, what I'm going to say is interphase. When the cell is not doing much of anything that you can see, there's still stuff there, but it's dissolved in the cytosol. So we've talked about the plasma membrane or the cell membrane already. It's a very important thing, and it's a phospholipid. The major structure of the plasma membrane is a phospholipid, and that should automatically remind you of what a lipid is and how lipids are assembled. The major parts of lipids most lipids can be triglycerides, and a phospholipid is a biglyceride with a phosphate group. The membranes around which the cell is organized, anything that I drew last week and anything that you see in this series of diagrams is a phospholipid bilayer, including around the nucleus. And the nucleus has a very, very important function, obviously, for the cell, but it's got a very, very well-developed phospholipid bilayer, and it's very strict on what it lets into the nucleus. And then the cytosol, or the cytoplasm, is the stuff that's inside the cell membrane, but outside of the nucleus. And it's got a series of little organs. Our body has, so our stomach, our small intestine, the large intestine are organs. The cells have little organelles. That suffix E-L-L-E means little. When you start talking about some of the characteristic functions of the cell, so what are some of the things? We talked a little bit about cell metabolism. Cell metabolism talks about all the different types of chemical reactions, and one or two examples of chemical reactions: dehydration synthesis if you're building something up, and hydrolysis if you're breaking something down. If I talk about hydrolysis, then which part of the cell are we talking about? Hydrolysis, those types of chemical reactions, not exactly, but those types of chemical reactions will be happening in the cytosol with glucose and in the mitochondria with pyruvate. Right? So that is an example of cell metabolism, and that would be one type of series of chemical reactions. And dehydration synthesis would be a different type. So that if you work out and you're trying to get bigger muscles, right, or you're trying to lose some fat, that illustrates two different things, right? In one example, you're trying to get bigger muscles. What type of chemical reaction are you doing? Dehydration synthesis. Where is it happening? It's happening in the cell. What is it called? Cell metabolism. What is the major macromolecule that you're making? Protein. If you're trying to lose fat, what is the chemical reaction you're trying to enhance? You're trying to enhance hydrolysis. You're trying to consume extra ATP. And in the process of consuming ATP, you're trying to get to the triglycerides that are resident in your body. If they reside in your body, they're there for a reason. And we'll talk about what lipids do when we get to chapter 4. So synthesis of molecules is one major thing, and when you see synthesis, dehydration synthesis is really the key thing. We haven't talked about communication yet. Communication occurs through a particular type of cell and a particular type of tissue. Does anyone know what that is? So nervous tissue. Tomorrow I'm going to talk about 4 different type of macromolecules. As it turns out, there's also 4 different types of tissues, one of which is neurons, or nervous tissue. And then finally, we'll talk about reproduction and inheritance. We're just going to gloss over reproduction and inheritance very, very generally. There's 2 different types of reproduction. There's only one type of inheritance. You inherited DNA from your parents, right? There was reproduction, cellular reproduction and biological reproduction to make you, right? And that's true. But since you were a developing zygote, you were a single cell. As you were a developing zygote, those cells then divided and they reproduced as well. So when cells reproduce and they're exact copies of yourself, there's one type of cell reproduction. And when you're trying to make a new individual, that's a different type of cell reproduction. That is called meiosis, and I'm not really going to be talking about that here. What we will be talking about, so that transmission of genetic information to the next generation, that's having a baby, and that's the next generation. Right? We're not what you're talking about. We're going to be talking about determining cell structure, cell function, cell location inside your body or my body, inside the individual, within a generation. So a human cell, I could also say an animal cell, because they're both the same. You can see this picture right here, and this is exactly what I drew last week, right? Obviously, it's not quite exactly. I don't think I got this much detail. But if you just look at this here, first of all, in the upper left-hand corner, there's a phospholipid bilayer. You can see all these lines right here, and these lines right here, and these lines right here, and these things right here. I showed you all, I drew more or less some of these same pictures here. And so if you were to look at this picture and then you look at your notes, they should match pretty well. If there's any problems or any errors in my drawings, then you please let me know and I'll fix them. Yeah?
When it goes to the cell, there's always just one nucleus in each cell.
So right now, yes, and I'm lying to you. Because when we get to other tissues, that's not true. But for right now, yes. You have a question?
Never mind then.
So the other thing I'm lying to you about is that all cells look like this, because they don't. But we're just going to say this is the model of a cell. And so when I say lie, it doesn't mean that I'm lying to you. Like, I don't know if you guys are listened to the political debate between Harris and Trump last week, but this person, Donald Trump, literally said that there are individuals that are practicing post-birth abortions.
Oh my God.
I was like, I literally said that to my wife earlier in the day, and she's like, "What are you?" And she swore at me, and she's like, "You're like kidding me, right? You're nuts, right?" So that's a lie, right? And by the way, a post-birth abortion means that's murder, right? And I don't know, the last thing I checked is like murder is not legal, right? So whether the individual's a day old, an hour old, or 100 years old, it's still murder, right? So I'm sort of, well, in that case, it's infanticide. Well, it might be infanticide, right? It might be so, but yeah, regardless. That's a lie. When I say I'm lying to you, right, I'm not. So this is definitely true, but it's not true for everything. This shape is definitely true. The fact that this picture only has one nucleus is definitely true. Most cells look like this. Most cells are similar to this. Most cells operate like this. So then when we get to other examples of cells that do something different, and we say, based on this, what's different? So that if when we go to muscle tissue, go back to this picture right here, and now let's go back, let's look at the muscle. What does a muscle look like? We go to nervous tissue. Look at this cell right here. How is it different? It's got a nucleus, it's got this, it's got that, but how is it different? Okay, so when I say I lie to you, it's based in truth, it's not based in, I don't know where. Anyway. So let's talk about, when I say organelles, the first and most important organelle is the plasma membrane. The plasma membrane is incredibly important. When I was talking to my son's roommate, he's an A&P too, I said, remember about the cell and chemistry from the first part of A&P 1? And he was like, sort of. And I said, well, it's going to come back to bite you on the ass hard if you don't. All about the structure of the cell membrane, the things that are called, in my work vocabulary last week, I called them integral proteins. They're going to change the names to something called receptors or ports, and there's a number of different terms that are associated with it. But they're going to let things in and let things out of the cell. Your stomach will let things in and let things out of the cell. Your small intestine, your large intestine, your kidneys, and etc. They are really, really focused on letting things in, letting things out of the cell. And that's all tied to the plasma membrane. The plasma membrane, we've talked about it enclosing and supporting the cell. I didn't mention much about extracellular. I did say ECF, or extracellular fluid. There's also something called the extracellular environment. The term extra means outside or beyond the cell. So anything beyond the cell is not inside the cell, and that's extracellular. Recognizing, communicating with other cells. I don't know why I'm talking about my son all day today, but last week he's allergic to certain nuts, particularly cashews, and he was at the store, at the school store, and he ate a breakfast bar, and he was like, I started feeling funny. Then I looked at the ingredients. He goes, it's a breakfast bar that I've eaten, but I've never had this variety. I know this model, right? This had cashew milk in it. And he chugged some Benadryl right away, and then he's like, half hour later, I started throwing up. That communication, that recognizing of stuff, that foreign information here, communicating with other cells, determining what's appropriate, what's inappropriate. In that case, his body said, well, whatever the hell this is, get it out. And so now his roommates are making fun of him. And determining what's moving into and out of the cells, what's that process? If you're going into a cell, it's endocytosis. If it's going out of the cell, it's called exocytosis. Those are general terms, and we're going to get more specific today. There's something that the cell membrane has, it's called a membrane potential. The membrane, so this, if you go like this, and this is say this is a membrane, there's nothing, you don't get shocked or anything like that, right? There's no attraction to this table. It's just there. It's just a physical barrier. But the membrane potential not only is a physical barrier, it's also got a chemical barrier, and it's attractive to some things and it's repulsive to others. And more than that, what's that called there? It's called hydrophobic, hydrophilic, and depending on where you are in the membrane, right, you can either be having things that are attracted to it and then repelling it, particularly within the center of the membrane. So there's positively charged ions, negatively charged ions, and I have to shout out to the cation, right, those cations and anions. So these are really important, right? They help determine what's going into and out of the cell. And the process that's called, or one process that I want to really focus on today, is the sodium-potassium pump. Sodium, not elemental sodium, there's ionic sodium, ionic potassium, sodium-potassium pump here is really critical, and that's the example we'll talk about today. So here's an illustration of the plasma membrane. We have ECF on the top, cytosol or intracellular fluid in the bottom. If that's true here, we have these blobs right here. These blobs, what did I call them? I called those integral proteins. And if we look more carefully, you're going to see another thing. There's a couple of terms here. So first of all, I'm not lying always, right? They are integral proteins. And an integral protein is a protein that's embedded in the phospholipid bilayer that goes from one side to the other. All right, now, there's different varieties of integral proteins. If you look in the center right here, there's something called a channel. Right? So there's integral proteins right here, and there's a membrane channel. And a membrane channel is another variety of an integral protein. So if you see that term channel and integral protein, they're the same thing. Later on in the semester, we're going to get into them, especially when we talk about the nervous system. Up on the top, if you look also on the superficial or the superficial portion of the plasma membrane, there's other things attached to it. And these other things attached to it, what do you think these molecules are? When I say these macromolecules, you can have a protein, nucleic acid, lipid, or carbohydrate. So don't say protein. Now this is a protein, but I'm talking about these yellow things.
Glucose?
So those, they might be glucose, but they're carbohydrates. Right? How do I know they're carbohydrates? Well, there's two things, two reasons. By the way, whenever I ask about questions in the answers, I mean from diagrams, if you look around, oftentimes the answer is in the diagram. So what are those yellow things? Those are carbohydrate chains. So I can read, I saw that already, but I also know the structure of monosaccharides, right? That six-sided figure here, that hexagon that I drew a couple weeks ago now, those tells me that those carbohydrate chains are what's something called receptors. And a receptor is something that sits outside in the hallway and grabs Rhini students and brings them in, if I had some, right? I don't have to do that because everyone's here. So there's going to be carbohydrates, and now there's not always carbohydrates, only carbohydrates, because there could be lipids, and then if you look up top, there could be proteins, right? So it's a little bit more complicated than that, but for right now, we're going to say there are these carbohydrates, lipids, and proteins that act superficially on receptors. They're tied to these proteins right here, and those proteins then act as, what's this thing, as channels, and these proteins are called integral proteins. We talked about the phospholipid bilayer, that phospholipid, that individual phospholipid. Why are these tails right here, some of them bent, and why are some of them straight? Because of the nature of the phospholipid itself, of the lipid itself, rather. So with saturated versus unsaturated. And then if you look at the internal, the cytoplasm here, the cytoplasm, and now I didn't draw this on the diagram, because what I've done in the past is if I started drawing the cytoskeleton, it just doesn't look good. It's already rough as it is, my drawings. I'm not an artist. But these things right here are called cytoskeletal elements. That's internal structure of the cell. And that basically, if there's a little bit of tension or pressure in the cell just doesn't disintegrate, or hopefully doesn't disintegrate. And then finally, when we were talking about lipids, we talked about different types of lipids, and we drew lipids, and then I said if you take it then to the next extreme, we can make very, very big molecules, one of which was cholesterol, and that cholesterol then can be derived, can make other things. Cholesterol gets a really, really bad rap for health in health circles, but without cholesterol, without lipid in particular, we would die, we wouldn't exist. So this is really, so there's a lot here, and hopefully it illustrates, it's familiar to you. Hopefully what we did last week is very familiar. So the plasma membrane here has some carbohydrates, lipids, and proteins here that And generally what those things do is they help act as receptors and they bring things into the cell, or they have as receptors inside the cell and they get things out of the cell. So those receptors there help to maintain the structure of the cell itself. I mentioned cholesterol just a minute ago. Cholesterol is interspersed within the cell membrane. It's really critical to that. So that, what type of molecule is that? That's a lipid. Interspersed among phospholipids. And that phospholipid nature of the cell is both, depending on where you are, hydrophilic and hydrophobic. And let's see. The book distinguishes between integral proteins and peripheral proteins. I don't, for the nature of this class. When you get into cell biology, and by the way, when I was talking about the structure of all the different enzymatic reactions in glycolysis and a citric acid cycle, if you are interested in that and you want to go into biochemistry, that's where you study the structure of those enzymes and what they do and how they do it, right? So that's beyond the scope of this. Peripheral proteins are important to the cell. There's definitely that, right? I don't really distinguish between integral proteins and peripheral proteins. I just sort of mush them all together as integral proteins. They more or less do the same thing. And if you look at the bottom here, the function of these integral proteins here, they can be markers, attachment sites, channels, a channel and a receptor or something very similar. They can be a carrier, a channel, or a receptor are all very similar. And then finally here, these integral proteins can act as enzymes. I haven't mentioned this earlier, but an enzyme that I already talked about, and I'll mention again today, is ATPase. That enzyme is bound to a membrane. In this case, this membrane it's bound to is not outside the cell, but it's inside the cell membrane. It's called the cristae of the mitochondrion. And that is bound to the membrane, and that helps generate ATP. So there's a number of different functions of these proteins here. And these proteins here can just basically recognize, they mark the cell, and they help cell functions. So whether you're talking about the cell membrane as an integral protein, or that's the protein there, or as a channel, it really doesn't matter. If you look at the picture on the left hand, or at the top here, here is the integral protein that's sort of got this particular binding site. It allows this thing on the left into the protein, but the thing on the right it doesn't allow. So it's specific. I don't talk about what's called the saturation of protein, or there's basically limits to this. And what happens with limits to this, and here's an example of things that we're going to talk about. I mentioned the sodium-potassium pump. The way that the cell works is that if the cell pumps out sodium and pumps potassium into the cell, but if there's a whole bunch of sodium floating around in the cell, right, and it pumps more out, right, then there's a lot of too much sodium outside of the cell, and what does that do? It has consequences for your blood pressure and other things. One second. So I don't talk about the rate here, or just all we're doing here is talking about the function of these integral proteins and the name where they're located. This is called endocytosis, importing into the cell. That's called exocytosis, exporting from the cell.
Never mind, you said you're not going to talk about the rate.
Is that?
You said you're not going to talk about the rate.
Oh, okay, yeah. Sorry, I didn't mean to kick you. I'm sorry about that. I thought that was a table. So I'm not gonna worry about the rates. Again, the rates then would be depending on the enzyme and the characteristic here. That would be something for biochemistry. So these transport proteins can be a number of different things. But if you look at the bottom, the transport protein that's most important, the one that I like to talk about the most, is ATP-powered pumps. And again, it's an integral protein, just like other transport proteins. We'll talk about other elements, but particularly for this quiz and for the cell chapter here, we're going to use ATP-powered pumps as one example. Again, these membrane proteins that are found embedded and go all the way through there can also be called channel proteins. Another thing, and now this becomes, when we're talking about the sodium potassium pump, another thing that they call channels here, and you can have different types of channels here. So if it, and the two major classes here, if it's a gated channel, it doesn't allow things in unless the gate gets opened, right? And the gate gets opened by that specific nature of the chemical. It's look for something, when that something comes in, the gate gets open. Or the channel leaks. It's like having a hose in your backyard and you just let it drip a little bit of the water coming out of the hose. So that's a leak channel. So there's a leak channel and there's a gated ion channel. And that gated ion channel, so there's two types of channels. They're all different types of proteins, integral proteins here. They're all different versions of this. And then there's different types of gated channels here. A ligand is a chemical and voltage is a charge. Ligand is a special chemical here. It's looking for a special chemical. That could be a hormone, for instance. That hormone, the cell receives that hormone and then it opens it up and then it does something. The cell starts doing something. The charge across the plasma membrane, one example of that charge, that voltage gated thing would be the sodium potassium pump. That sodium potassium pump, when you allow the sodium to move into the cell, or potassium to move out of the cell, that changes the charge of the cell, and that voltage gated channel then responds. One example of problems with this sort of channeling and the channels that don't operate is called cystic fibrosis. It affects the chloride ion channel on the lower right-hand corner, the chloride ion channel, and as a result of that, it causes thick, viscous mucus to be produced by cells, and it's all because of the channels not operating. So in this case, now I mentioned sodium potassium channels. Here, this is a chloride channel. Chloride channels then, when they don't operate, it has consequences for the body. I mentioned sodium potassium channels. Here's the first time that I'll show you this. The sodium channel pushing. So in this picture now, this pink right here, these pink ions are the sodium, and these purple ones are the potassium. Now, if you look at this part right here, where my pointer is, that pointer, that's the ligand gated, the ligand channel is closed, that gated channel is closed to sodium. Right? And what happens to potassium? Well, nothing. Right? It doesn't allow potassium at all. On the right-hand side here, the ligand channels they're looking for something specific. So these two things are the chemical, the ligand that it's waiting for. When that thing arrives, then it opens. So that's a key to the lock, basically. So that ligand is a key to the lock, and that opens up, and it lets something specific. On the left-hand side is a leak channel, and that leak channel allows for things, in this case, potassium to slowly leak out of the cell, and then it gets imported into the cell under appropriate conditions. There's also another term associated with this called transporters, and transporters basically help this stuff gets across this phospholipid bilayer. I'm not going to talk about different types of carrier proteins here, in the uniporters or symporters, the antiporters. I'm going to distinguish there. But I do talk about ATP powered pumps. So now if you go back to your notes here, you should automatically say, oh, I know what this is. This is in the cristae of the mitochondria, and he drew the mitochondria with that little wiggly thing in there. Oh, that thing is a phospholipid bilayer. Right? I didn't feel like drawing that phospholipid bilayer there, right, in the cristae of the mitochondria, but that's what it is. And here's what happens here. So that we have 3 steps right here. The ion is attached to the, this is the integral protein, and the name of this integral protein is the ATPase. All right, so I didn't put that down. So anytime you see a molecule with ASE, then it breaks that down, or it does something with that. ATPase is an enzyme associated with ATP. Okay, so that's the function of this. Now, if you look at this ion right here, it's on one side of the phospholipid bilayer. Because that ion is charged, and the nature of it has got to be charged. If you look at the bottom here, it's moved to the other side. Now, what happens here during this? Now there's oh, wait a minute. I'm mixing up. I'm jumping way ahead forward in the mitochondria. This is how the cell makes ATP, uses ATP, not makes ATP. I messed up. So ATPase definitely makes ATP, okay? That is absolutely true. I'm jumping ahead. I thought I had done something different here. What we're doing here is we're going from ATP to ADP. The principle is still the same. The ion is on one side of the membrane. It has to get to the other side, and in the process, it requires energy to do that. How does it get across there? Where does the energy come from? The energy comes from this ATP. That ATP then provides the power to push it across that membrane. So as it pushes it across there, that ATP then becomes what? It becomes ADP plus what? Plus inorganic phosphorus. If you go back to when we were talking about assembling ATP, I did this chemical reaction in the mitochondrion. I did ADP plus inorganic phosphate, and I said it's this. I said it goes both ways. It goes left and right. It's a bidirectional. It's a reversible chemical reaction. What do you mean ATP? So in this slide then, we're talking about what's called the hydrolysis of ATP. That means that it's breaking down ATP. So that ATP now separates here. That P right there, that inorganic phosphate, is no longer attached to the ATP. And then the ADP and the inorganic phosphate separate. What happens to that then? That gets transported to the mitochondrion, and then you assemble, and then you go from left to right. Receptors generally are on the outside of the cell in this extracellular fluid, and they determine what's going to be going into the cell or whether or not the cell should react to something. When my son ate that cashew milk, right, that stuff that he's allergic to, your cells had receptors. They received those receptors. His cells, not your cells. His cells had these receptors, and they said, there's emergencies here. Let's get rid of this, whatever the hell this is. And then he threw up. Generally speaking, those receptors are linked to channel proteins. And again, those channel proteins are integral proteins. And here's, I'm sort of belaboring this point now. These sodium channels right here are important. There's another molecule here. This is called a neurotransmitter called acetylcholine. Acetylcholine is incredibly important. It's important in muscle functioning and also nerve functioning, neuron functioning. So acetylcholine is one example of a receptor, a ligand-based receptor, and it's linked to the sodium channel. So now the sodium channel, what does the sodium channel do? It allows for sodium to pass into the cell or out of the cell. depending on the particular nature of it. I've talked a lot about getting stuff out of the cell and into the cell, and what's that called? It's called cell transport. Cell transport occurs when the cell membrane, the plasma membrane, allows only some things in and not other things in. This class is an example of a selectively permeable class. Right? I don't let all URI students in here. I only let certain URI students in here. You're all members of the URI community in addition to the Rani community. So that's selectively permeable. It only allows certain things to go in. And what are some things that it allows to pass through? What are some things that it prevents? When there's something in high concentration in the cell cytosol or cytoplasm, the cell doesn't allow that stuff to get out. So then there has to be special conditions under which it allows it to get out. Sodium, calcium, and chloride are higher concentrations in the extracellular fluid. All of these things are either large or chemically charged. In all these cases, it doesn't allow it to get in, but sometimes you need that stuff to get in the cell. So those chloride ions, you need it in the cell, that sodium and the calcium. You need it into the cell under certain conditions. Another thing that happens is that the cell has to stay more or less the same size. Right? You don't want it losing a whole bunch of water and never replenishing it. That's called dehydration, right? When you lose too much water, not dehydration synthesis. If you get too much water, right, that could also allow the cell to burst or cause it, and we'll talk about that later on today. So if it's chemically large, like a protein or glycogen, if it's chemically charged, then it's either going to keep it out of the cell or in the cell, depending on where it is. If it's uncharged and small and lipid soluble, so there's oxygen and carbon dioxide, those pass through the cell membrane easily and without any control. And then again, if it's large and it's lipid or non-lipid, if it's large, it has to get a transporter. And what's that transporter called? An integral protein or a receptor. So in all cases here, we're talking about movement across that phospholipid bilayer. And when we talk about movement across that phospholipid bilayer, I mentioned one example already without telling you what it was. But you're talking about either moving it actively or passively. So passive movement, passive membrane transport has 3 varieties. Active membrane transport has 1 variety. Active membrane transport is using ATP, and it goes from without it, it doesn't happen. Using ATP, it goes from this, it breaks up, it hydrolyzes the ATP, it breaks up the ATP, and in the process it makes this, and then it uses the energy in that last chemical bond between the phosphates here. It uses that to allow things to cross the phospholipid bilayer. So that active membrane transport is pretty simple. And I'm going to use one example of that in a minute, and I'll talk about the sodium potassium problem. At the bottom of this slide here, it's called vesicular transport. What is this term vesicle? Right? And if you look back at your notes here, I drew a vesicle. I drew a membrane-bound structure. If that membrane-bound structure is inside the cell, then it's going to get out of the cell, and that's called exocytosis. If that membrane-bound structure is outside of the cell and is trying to get in, that's called endocytosis. Let's talk about passive diffusion. Passive membrane transport. The first example is diffusion. Diffusion occurs when there's high concentration. In all these cases, you're moving stuff from an area of high concentration or low concentration, or from an area of low concentration to high concentration. This doesn't happen if you have the exact same number of ions on one side of the membrane and the other. Nothing happens. There's something called net transport. In order for net transport to happen, you have to have a whole bunch of stuff on one side and less on the other. When you have a whole bunch of stuff on the one side and less on the other, and these are small molecules, oxygen and carbon dioxide are two examples. Then they move from an area of high concentration to low concentration. So that when there's high concentration of oxygen in your blood, a low concentration of oxygen in your cells, where does oxygen go? Into your cells. Conversely, the opposite is true too. High concentration of carbon dioxide in your cell, less outside of the cell. Where does carbon dioxide go? Outside of the cell. It doesn't happen without this concentration gradient. If there's no concentration gradient, then there's no net movement here. So this low to high concentration or high to low concentration is important. When it's diffusion, it goes from high concentration to low concentration. When it's passive transport, from high concentration to low concentration. Charlie?
Would they just go back and forth, going from high concentration to low concentration?
So for diffusion, it always goes high concentration to low concentration. It's never the opposite. Here's an example of that. And back when I used to teach chemistry and biochemistry at Rhine, I would do this example here. Get a block of salt or a block of colored stuff, put it in water, and slowly but surely it moves away from where it started. If you look on the left-hand side, there's a high concentration of stuff right here, and there's a low concentration of it away from that. And so what happens here is that the ions then move away from the high concentration. There's a number of things that affect the rate at which that, but the thing that's most important is the concentration gradient. So there's some things that are in this picture right here. There's some things that pass through with the cell that doesn't, it's not regulated by the cell. Other things that are regulated by the cell. If you look at this diagram right here, here's something that's regulated by the cell. It's bounced off the cell membrane. Here's another thing that's regulated by the cell. It's allowed to get into the cell through the membrane channel. And then finally, these yellow things are going from outside the cell to inside the cell because there's more stuff outside the cell than inside the cell. Another thing that is passive transport and is incredibly important is water. And that when you're talking about the passive transport of water, that's called osmosis. Osmosis passes through that phospholipid bilayer. Generally, there's also these things called channel proteins called aquaporins that help for the movement from high concentration to low concentration. But if you have a lot of water in your body and the cells are saturated, there is enough water, right? Then the water, you're going to try to get water out of your body through a number of mechanisms. Right? If you have too little water in your cells, you're going to try to get water into those cells. How does it get into those cells? Through osmosis. How does it get out of those cells? Osmosis. How much energy is required? Zero. Right? I don't really talk a lot about osmotic pressure, but I will illustrate what happens to water moving into and out of the cells through these next couple of slides. So there's something called osmotic pressure here, and that when the chemical concentration of the cell, outside of the cell, is different than inside the cell tries to, the water moves. All right. So there's three, excuse me?
Is that also called like isotonic?
So isotonic, hypertonic, and hypotonic.
Yeah.
Right. So there's this isoosmotic, and you look at this prefix iso, hyperosmotic, hypoosmotic. Iso means the same, regardless of what you're talking about here. Hyper means that it's greater on one side, so that if you have if you know a kid that's hyperactive, he or she has a lot of activity. Hypo means less. A hypodermic needle. A hypodermic needle is a needle that goes into the body and it goes hypo the dermis. Where's the dermis? It's deeper to the epidermis, so it goes deep to the dermis. And Finn, here's what you were talking about. Isotonic, hypertonic, and hypotonic are conditions for three cells, and the cell that we're going to talk about are the red blood cells or erythrocytes. So when you take an individual erythrocyte and you place it in an isotonic solution, a hypertonic solution, or a hypotonic solution, what happens to the cell. Again, now this is talking about the cell volume and without any energy required. The cell has water residing in it. So at the top there, the cell, that red blood, those erythrocytes have water inside. You put it in one of these three solutions here. And if you look in the middle here, this cell, this picture, and the top picture, they look the same. The red blood cell hasn't changed structurally in size, in shape, in volume. And if you look to the left, it's gonna blow up, and basically there's too much water going into the cell, and if you look to the right, it creates or shrinks. All the water has left the cell. So when there's a whole bunch of salt outside of the cell, or when there's very little water outside of the cell, and there's more water inside of the cell, what happens? The water goes out of the cell. If we were adrift on a boat in Block Island Sound, in Narragansett Bay, and we drank salt water, and we drank it for a long time. Why are you looking at me like that? And we drank it for a long time, what would happen to it? The salt water in our body would go into our tissues, right? It would go adjacent to the cells, and eventually that's what happened, right? And that would be bad. Right? If we kept doing that for too long, we would die. Why? Well, the cells, red blood cells, erythrocytes, and other cells would start losing water so dramatically that we wouldn't be able to function properly. Conversely, if there's too little salt in the water, and there's too much water out there, the water pushes into the cell. That's a hypotonic solution. It pushes into the cell, and ultimately, if there's too much water going into it, the cell can burst. One last thing on passive transport here, and so here's a picture of that. Facilitated diffusion. Diffusion is the passive movement of stuff from high concentration to low concentration. Sometimes it can have help. When there's big things, a carbohydrate, for instance, when there's big things and you try to get it across that phospholipid bilayer, but the phospholipid bilayer doesn't allow it, then you have to facilitate it. You have to allow things to get out and get across that phospholipid bilayer. What do you use? A channel protein or a carrier? What's that also called? An integral protein. What happens here? There's a receptor associated with that thing, like a carbohydrate. That receptor bonds to the protein or the integral protein, and that protein then allows it to go from one side of the cell to the other, all through concentration gradient. So from there, high concentration to low concentration. So facilitated diffusion, I also use the term receptor-mediated endocytosis. That term, receptor-mediated. What is a receptor-mediated? So it's receptors that are outside of the cell. Receptor-mediated endocytosis. What's endo? Going into the cell. Endocytosis, getting stuff into the cell. How do you get it into the cell? Through that receptor. Facilitated diffusion and receptor-mediated endocytosis for this class are the same thing. So in this case now we're just talking about glucose getting into the cell. And now what I want to do right now is I want to take a break. It's 9:44, so it's 9:45. So let's take a 10-minute break, please.